Hello, General Biology, and welcome to the second in the screencast series for Section 2.4 of Chapter 2, and we were just talking about energy changes. And just to refresh your memory in case you listened to the other one and it was a little while before you're getting to this one, we were talking about energy changes, and energy changes happen when bonds are broken and bonds are made. And when bonds are broken, we're going to release the energy. When bonds are made, we're going to store the energy. So when they're broken, we're going to call that an exergonic reaction. And when they're formed, then we're going to call that an endergonic reaction. Okay, so here is an example of an energy releasing reaction. And if we take hydrogen and we burn it, hydrogen gas, the hydrogen will react with oxygen and it will produce water vapor. Alright, the energy is released in the form of heat, and sometimes when the hydrogen gas explodes, and the light and the sound. So when you burn something, you're burning a match. That is an energy releasing reaction that's taking place. Okay, if we're going to then take hydrogen and oxygen and we're going to make that water vapor, then this product from burning that hydrogen, that energy release, is going to give us water, and it's going to bond the hydrogen and the oxygen together. So we will get a little bit of energy that's stored in the hydrogens and the oxygens there. The reverse reaction, in which water is changed into hydrogen and oxygen gas, absorbs so much energy that it generally doesn't occur by itself. So if we need to break this down and get hydrogen and oxygen, this is going to have to take an awful lot of energy. We have to, and I won't talk about Celsius, but we have to, if we want to turn water into gas, we've got to first boil it at 212 degrees Fahrenheit or 100 degrees Celsius, and then we're going to have to take that further in order to separate the hydrogen and the oxygen gas. So it's going to absorb a lot of energy before it actually breaks down. So the only practical way to reverse the reaction is to pass an electrical current through the water that actually decomposes or breaks down the water into hydrogen gas and oxygen. So, as it says here, in one direction the reaction produces energy, and in the other direction the reaction requires energy. So notice it says note, do balancing equations here. I'm going to make a note, and that's what we're going to do in class for one of our activities. So there's different forms of energy that are used in order to carry out the chemical reactions to stay alive. Plants are going to get their energy by trapping and storing the energy from sunlight, and they're going to make energy-rich compounds like glucose, which we're going to see in the process of photosynthesis. Animals are going to get their energy when they consume plants or other animals. So those food molecules that we take in, when their bonds break down, that's where we're going to get our energy from. Humans are going to release the energy needed to grow, to breathe, to think, to listen to Mrs. Saunders and these wonderful screencasts. Dream that you're not listening to Mrs. Saunders doing one of these screencasts. And all of this is through chemical reactions that occur when we metabolize or break down our food that we are eating, our digested food. So those are some of the energy sources. Hear that bird in the background? That's a blue jay. I'm outside on the front porch. So the energy needed to cause a reaction is called activation energy, because some reactions do not always occur spontaneously. We need some sort of energy to start the reaction. So if I was sitting at the top of a hill and I had a basketball, that basketball could sit there at the top of the hill and not do anything. It needs a little push, a little force to get it rolling down the hill, and that's the activation energy. So sometimes that's needed within the reactions. So let's take a look. These are graphs that show activation energy and how activation energy works. As it says, and I'm going to read this to you, the peak of each graph represents the energy needed for the reaction to go forward. And then the difference between the required energy and the energy of the reactants is the activation energy. And then activation energy is involved in chemical reactions whether or not the overall reaction releases or absorbs energy. So here's our reactants. Here's the peak. This is the energy that's needed to get the reaction to happen, and it causes the products to be produced. Because this is not a steep change here, this is an energy absorbing reaction. So that means that we're going to have bonds that are going to be formed, and that energy is going to be stored. Where in this one, the reactants go up, we get the reaction, the activation energy needed, and the reaction continues, and we've got a steep drop, and this is an energy releasing reaction. So let's go back to those two terms. This is going to be an endergonic reaction, and this is going to be an exergonic reaction. All right. I want to go back. Well, this is a good spot. So most of the time, activation energy is in the form of heat. And guess what? Heat is deadly to living systems. I mean, we don't eat a hamburger and then turn on the stove or the oven and jump in and help. That's how we digest it. That wouldn't be a good, healthy thing for us to do. So not all reactions can happen as a result of heat. We've got different forms of activation energy. Heat is the main one, but we can get light, we can get sound, pressure can create a reaction. Temperature, that's heat. Those are the main ones. Light and sound and pressure. We can have radio waves as well. Okay, so there's lots, which kind of goes along with sound, but these are the different ways that activation energy can come in, the different forms. But let's focus on heat, because sometimes, as I said, heat can be damaging to living systems. So we have to lower that activation energy in terms of heat. And we do that by a special protein called enzymes. So we're going to look at the role that enzymes have in living things and how it affects their function with the chemical reactions. So sometimes chemical reactions can be too slow. They don't happen fast enough. They don't have the activation energies that are needed, or the activation energies are too high. So they need something that is more practical for the living tissue in order to go through the chemical reactions. And in these cases, we use these specialized enzymes that are called catalysts. And a catalyst is a substance that speeds up the rate of a chemical reaction. So you're familiar with this. Have you ever had your nails done? Nail polish? Sure. And you know that strong odor? That's acetone. Acetone is actually put in the nail polish. It has a high evaporation rate, and when it evaporates, it dries the nail polish. Or now you get those gels and the ultraviolet light that you put your hands under, that light source. That is the activation energy needed to dry the gel nail polish. But these are catalysts. Well, enzymes are catalysts of living things. And the enzymes in human systems, they lower the reaction's activation energy. They lower the amount of heat, they lower the amount of sound, they lower the amount of light, they lower the energy needed to cause a reaction to start. So enzymes are nature's catalyst because these are proteins, they're specialized proteins, and they speed up chemical reactions that take place in cells. And they do this by lowering the activation energies so that the reactions can happen. So let's take a look at this other graph, and you'll probably see graphs similar to this on the test. So here's our reactants, and if we have no enzyme, it takes a while for the reaction to build up and then the activation energy to cause it to go down. But if we have an enzyme, it doesn't have to go up quite as high, and then it can come down to produce the products that we need. So that's how an enzyme is going to work. It's going to lower that activation energy. We don't need to go to this point if we have an enzyme. We can go to this point and still have that reaction occur. Okay, so let's take a look. We already talked about this equation in the beginning, and it says, for example, the reaction in which carbon dioxide combines with water to produce carbonic acid is so slow that the carbon dioxide might build up in the body faster than the bloodstream could remove it. So your bloodstream contains an enzyme called carbonic anhydrase. And this, anytime we see ASE at the end of a word, it means it's an enzyme. So this carbonic anhydrase speeds up the reaction by a factor of 10 million so that the reaction takes place immediately and the carbon dioxide is removed from the blood quickly. Okay, so that's what that does. We're going to do a lab on enzyme activity, and we're going to use liver. I'm going to get chicken livers, and we're going to use hydrogen peroxide. And there's catalase. There's a special type of enzyme inside the liver, and when you put the hydrogen peroxide, it's going to bubble and it's going to fizz to show you that the reaction is happening. Enzymes are very specific for a reaction. They generally only catalyze one chemical reaction. So, for example, protease is an enzyme that breaks down protein. It's a digestive enzyme. It's specific only to proteins. There's another one called amylase. And see, we're ending in ASE here. Amylase breaks down carbohydrates. Specifically monosaccharides. So the enzyme is specific and can only work for that one reaction. So carbonic anhydrase gets its name because it also catalyzes the reverse reaction that removes water from carbonic acid. So it can be used both ways in the reaction to make, if we go back to this, it can be used to make the carbonic acid, or it can be used to take that carbonic acid and break it down into hydrogen, or water, sorry, hydrogen, I want to say hydrogen, and carbon dioxide, water and carbon dioxide. Okay, we're going to stop here, and in the next screencast we're going to talk about the enzyme substrate complex. Okay, see you in a few.